Okay, so I'm going to start. It's 9 a.m. in Southern California on a gloomy morning. It's 6 p.m. in Brest in France, where I gather some people from the chat. Some people are already set with for a session of beer and DEB. And I'm going to, in this second week of the class, attempt to say something about three themes. I'm going to talk a little bit about the strengths and limitations of models that simply take account of biomass in the way I described last week. I'm going to introduce the concept of energy reserves, both in a rather general way and in the very specific way that it's used in Bas-Kooijman's DEB theory. And I'll be doing the second as part of a general introduction to Bas-Kooijman's DEB model, a introductory material which will carry over into next week's class. Let me start by reminding you that in last week I set up some formalism for describing models of growth and reproduction of individual organisms based on considerations of biomass balance. And in that lecture, I developed a mechanistic representation of growth and reproduction in different food environments. There were a few side issues as we went along, but one that will recur throughout this lecture, towards the end of this lecture, and get a lot of emphasis in the next one, was that even there we introduced a rather powerful interpretation of respiration as being a combination of the costs of maintaining an organism plus the overheads on various processes that were going on. Although I'm not going to talk about it in these lectures, the formalism I described last day can be extended to include a description of sublethal effects of contaminants, obviously relevant to the CEIN people that are watching. The response to more complex food assemblages, you just change the functional response part of the model. And effects of food quality. I mentioned at the start stoichiometry. I haven't actually shown how to incorporate it. And at the bottom of the page, I've given references for each of these. Finally, the biomass budget models connect very naturally with the large body of ecological theory on size-structured models, for which there is a remarkably good book that has just appeared in January 2013 by André de Roos in Amsterdam and Leonard Persson in Umea, one of the titles in the Princeton University Press series in a population and community ecology. It's a fabulous book, and if you're interested, you can follow that line. However, these models have very substantial limitations. Perhaps the most fundamental is that I set it up describing an animal which got energy inputs from food and used the energy for food for various purposes. But some life stages of pretty well all organisms don't feed, and an obvious example is an embryo, in which a large amount of development and physiological change occurs with no input of food, and another example would be the pupal stage of an insect. A second reason why these models are highly limited in their applicability is that an organism is characterized by a single variable, namely its size or its mass. And yet there are many, many examples where organisms with the same size or weight may perform very differently in the same environment. And I'm actually going to elaborate a little on these, but I'm going to take examples from a nutrient-limited growth of microalgae, from starvation recovery of a small animal, and from a growth of a fish. Another feature that biomass budget models don't take account of is the fact that energy is not allocated to reproduction in a nice continuous way. Reproduction tends to occur in a series of bursts, and in many cases the allocation of energy to reproduction does not proceed in the uniform way that was described in that model. Taken together, these limitations amount to recognition of the fact that the biomass budget models, though they're powerful descriptive tools, take no consideration of metabolic organization. Effectively, they say an animal is a bag of carbon atoms, and there are certain rules for how they're utilized. They don't take account of any of the central organizing features of physiology, and that's obviously where we want to go when we're trying to develop DEB theory. Let me elaborate just a little on the examples. I'm starting with one which has a bit of historical significance in the context of DEB theory. The graph there shows the growth of the population of a unicellular alga, Chlamydomonas reinhardti, on the continuous curve, and the data points that I'm tracking here represent the amount of nutrient in the container where it was growing. The message is that population growth continues long after absorption of nutrient stops. And the interpretation of this is that the organism stores nitrogen from the environment. It gets energy from photosynthesis. All the time here, there is no input of nutrient, yet the growth rate is clearly changing. We need more than one variable to characterize what's going on there. Another example from my favorite animal is a rather old study, and the reference to the Bradley paper is given 2 slides back, where Daphnia were grown, and at different points in the life cycle were introduced to either a short period of starvation as shown there, or a longer period of starvation as shown there. Changes in the pattern of reproduction and of growth were measured, and I just want to highlight one feature from these data, which is that if you look at the changes in length, then there are organisms of the same length here, which according to the rules of our previous model would imply they would be growing at the same rate, yet these are growing slowly, this one is growing much faster. So individuals with the same length and food environment grow at different rates. We clearly need some additional information in order to describe that. And I also note, because it becomes relevant in the next slide, this idea of compensatory growth. There is a large amount of literature on growth in different types of environment in the context of aquaculture, and I've just shown one example here because it was a study which specifically included estimation of the von Bertalanffy growth rate, which I talked about in the last lecture. And on the x-axis here is the rate at which the fish were being fed. On the y-axis is an estimate of the von Bertalanffy growth rate, the quantity that I called gamma in the last lecture. And this quantity varies with food density. According to the biomass budget model that I put together in the last day, it should be invariant. It should not change. The details of how it changes are slightly surprising in this context, but the key message here is that it changes. And from that, we conclude again that this type of model with one variable characterizing the state of an animal is inadequate. And the obvious way around is to introduce some second variable that characterizes the state of an animal. And in each of these experiments, it's reasonably clear what that might be. In the microalgal experiment, the reason for the phenomena, for the pattern that we saw, is that the cells are capable of absorbing inorganic nitrogen faster than they are able to use it to support all the processes that lead up to cell division. In other words, the rate at which it's absorbing nutrient is much faster than the rate at which cells can divide. This results in a build-up of surplus nutrient within the cell, and once they run out of food, they're capable of using that surplus nutrient to support more growth. In the case of the Bradley et al. Daphne experiment, there are many interpretations possible, but there is a lot of evidence from these and related experiments to changes in energy allocation priorities after starvation, and obviously during the period of starvation the animal was subsisting on energy reserves. And in the sea bream example, there was again evidence of some changes in energy allocation priorities after a bout of starvation. I mean, taking it all together, it says that trying to describe what's going on with just one variable doesn't cut it, and we need something more elaborate. And the box at the bottom just really just repeats what I just said, that we need to recognize two types of biomass in any context where we want to describe growth and reproduction of an animal in changing environments. The fact that energy reserves are clearly part of the story is recognized in many subdisciplines where people use a condition index, which is essentially the ratio of the weight of the animal to the cube of some measure of the length, as a measure of this variability. But that's not a mechanistically useful thing, except as a derivative of quantity. We want to get to some more basic ways of thinking about it. And to do so, we need to actually grapple with this concept of energy reserves and come up with some definitions. And if you go through the literature, you'll find a whole range of different types of definition of of energy reserve. But you can characterize them, I think, in two groups. One is what I've called a chemical characterization, and the other is what I've called a functional characterization. The chemical characterization looks at groups of compounds like fats, lipids, certain proteins, or it looks at stores of elemental matter like the nitrogen I was talking about before, or it looks at it lumps the energy-rich compounds into a set of still defined and identifiable compounds that are regarded as, in some sense, metabolizable. The alternative is to characterize reserves in a functional way, and one which is, again, rather narrow, is to define reserve as biomass that is capable of supporting reproduction. That's a material in an organism that will eventually work its way to reproduction. But the most general way and the most useful way that is used in Bas Kooijman's book is to think of biomass as that part, think of reserve as that a part of biomass that does not require maintenance. And I've referred there to the Kooijman 2010 book. Virtually everyone that uses it refers to this as DEB 3, and subsequently in the lectures, if you hear me use the phrase DEB 3, I'm referring to that book for which the full reference was given earlier. Let me say a little more about these models that do work with chemically characterized reserves, both to show you something of the strengths and limitations. I think the best example and the example that's still most used is probably the group model of algal growth, which is a historic interest because it is the precursor of DEB, and Bas Kooijman has said that it inspired much of his thinking that led to the development of the DEB models. In this model, reserves are defined in terms of key minerals, and in the example I had before, I was thinking about nitrogen. All rates, that's rates of assimilation, rates of cell division and so on, are assumed to be proportional to cell volume. That's what I called volume assimilation in the previous lecture. Okay? This model quietly ignores maintenance costs. Growth rates are assumed to depend on the ratio of carbon to nitrogen or carbon to whatever is the limiting nutrient in this model. The parameterization of this model is very straightforward, especially if you have data from chemostats, and it is widely used to describe phytoplankton. I've given a reference at the bottom of the page to a particularly good book that uses this and related models. It's the book on resource competition by Jim Grover, but the early chapters of that book give a particularly lucid explanation of this type of representation. Some models go much further and characterize reserves in a way that takes account perhaps of both chemical considerations and some anatomical considerations. And I've illustrated in this picture the scheme used by Tim Planchek and several collaborators, including myself, in a model of in the model of the northern right whale. And this model took account of a large number of factors, but I highlight the fact that there was a component that represented energy-rich compounds in the blood. There was a component that represented structural biomass, which is a concept that's going to recur later in this lecture. There was a separate component that represented structural lipids, a distinctive feature of marine mammals, and then there was a lipid energy storage. And I've summarized the features of this model. It had lots of state variables. It had two types of reserve, the lipids and the structural lipids. It had explicit representation of a transport medium. It had very complex rules for the onset of reproduction. It included energy for fetal development and lactation. It was coupled to a toxicant model, and it was used for certain very specific predictions. There is no way you could build a general theory around a model like this. Equally, if you have one or two very focused questions, like the example at the end there, it's a useful model. But broadly speaking, these models have their limitations. And they raise conceptual issues. There's a large range of organisms that have broadly similar physiology and biochemistry. The biomass mudgid model that I talked about last day had a representation in which the von Bertalanffy growth rate, the rate of approach to the maximum size, was related directly to the cost of basal metabolic rate. Many animals, from the smallest to the largest, have pretty much the same processes involved in energy metabolism, and yet the von Bertalanffy growth rate varies hugely. The figure on the left has its x-axis, a measure of the size of the animal. It's volume on a log 10 scale, so it's a big range of variation. The y-axis is the von Bertalanffy growth rate, and again, on a log scale to the base 10, there is a lot of variation there. We would not expect the basal metabolic rate to vary that amount if there is pretty much the same physiology going on across many of these animals, which says that the representation in the simple model is not ideal. And there is a clue if you look at data on the variation of respiration rate. Again, a log scale, this time body weight on the x-axis, a measure of metabolic rate in watts on the y-axis, again on a log scale. And the data actually fit, as is well known, a allometric relationship with a, depending on which subset of the data you use, an exponent somewhere in the region between 2/3 and 1, with some people liking a popular number of 3/4 in between. But that just begs the question, why the difference? So there's indirect clues in terms of organismal performance that we need more than just biomass, in addition to problems in matching experiments to in matching data to theory. So to summarize up to this point, the empirically based models, as well illustrated by the whale example, have huge data demands. That in turn means that there are problems transferring them to new systems and different environments where you need more empirical relationships, and the last slide gave the example of the body size scaling relations. If you're interested in toxicology, you may have encountered what are often called QSARs, quantitative structure-activity relations, that tell you how differences in contaminants and in target organisms tells you about that variation, analogous to the respiration plot that I just put up. And the question that I want to use the remainder of this morning's sessions to address and to answer positively is whether some kind of general theory can help resolve these problems, or are we still working with essentially a large number of context-specific models? And if we are developing general theory, we need to take a somewhat different approach from the approach I've taken up to this point. That is, we need to identify some taxon-independent characteristics of organisms. That amounts to saying that the model formulation will be more abstract than in the models I've talked about so far. I've tried to be very concrete especially in the last lecture, in the variables I introduced, what they mean, how they relate to experiments, and so on. If you want a description that works from organisms, it works for plants, animals, and microbes, works with animals from Daphnia to blue whales in size, we're going to need some more abstract concepts. And the approach that has done this way better than any other that I'm aware of to date is Bas-Kooijman's dynamic energy budget theory. And this theory takes a very abstract approach. We're going to try and develop some of these abstractions. And to evaluate it, I think it's worth our while carrying two thoughts in our head. One is that general theory is really powerful for the reasons I've just described. It potentially lets us get around a lot of the difficulties I've been describing, but it's dangerous in the sense that if the premises are wrong, it lets you extrapolate further from observation, and so we are playing with something that is potentially very powerful and yet challenging to use. I'm going to present Bas-Kooijman's model in detail in just a couple of minutes, but I first want to present a slide that I've characterized as a sloppy representation of the Kooijman model in order to introduce what are probably the two key concepts for understanding the theory, and these are the concepts of structure and reserve. The Kooijman model, as applied to an animal, like the models we've been dealing with before, imagines that energy or carbon is absorbed from food, but it's then assumed to enter a compartment, if you like, that is called reserve. And it's not accident that the S has dropped from the word reserves that I've been using here in in an attempt to separate this somewhat abstract concept from the reserves that are commonly used in lots of other areas, people writing in depth theory tend to drop the S. And the distinction is the distinction I mentioned a little bit earlier. That is that structure or structural biomass is biomass that requires maintenance, and reserve is everything else. It is biomass, it is specifically biomass that can be mobilized for metabolic processes. And metabolic processes include making new structural biomass. It includes anything associated with energy metabolism, hence respiration, production of metabolic products. It includes fueling eggs, anything else that goes towards reproduction. And the formal development of this model is essentially the subject matter of the second chapter of DEB 3. And I'm going to talk through the various steps in setting up this model in some detail. But it's useful, before we get into the detail, to capture this idea, this broad energetic scheme, before we start thinking about subtleties. Energy comes from food. A certain amount is immediately excreted. It goes into a reserve compartment. From this reserve compartment, it is utilized for various functions, which are those that I have highlighted down the right-hand side of the slide here. Before we write down the model, there is one concept that I think is critical in understanding the theory, and for me represents the most important part of the abstraction. And that is that reserves and structure are what are commonly called generalized compounds. They're mixtures of large numbers of compounds with constant chemical composition and constant thermodynamic thermodynamic properties. So reserve is not lipids. Reserve is not certain proteins. Reserve is some mixture of a whole range of compounds. Likewise, structure. The theory is tractable and develops testable predictions because generalized compounds are assumed to have an unchanging composition. That's to say, the makeup of reserve in any one context, for any one organism, is assumed to be unchanging. The makeup of structure is unchanging. Of course, the relative proportions of structure and reserve can change. That's the benefit of having a second variable. In a few examples, we have to be more explicit than that, and generalized compounds are represented stoichiometrically by formulae of the form that I've written here, a some mix of C, H, O, and N. And by convention, these are written down in a way such that the index of carbon is 1, which actually makes a very nice connection with the way I worked in the previous lecture. And these indices X, Y, and Z represent the proportions of other elements relative to carbon. And the DEB book gives certain values as representative for some microorganisms. There is more detail in a paper that I actually forgot to cite at the bottom here by Tanya Souza, not the one that's cited there, which is relevant for a different reason, but a paper published in Physical Review. A useful unit that you will encounter if you read the DEB literature for working with generalized compounds is C-moles, and this matches very nicely again with the way we were thinking before. You can think of the model in terms of the flow of carbon in C-moles, and that is the way I'm going to present it. A C-mole is just the carbon mass of in any compartment expressed in moles of carbon. And if you want to convince yourself or to test whether you understand the concept of C-mole, simply do this little exercise, and I'll mention the answer to that at the start of next week's lecture. One of the impediments to understanding Bas-Kooijman's DEB model is the rather large amount of notation that is involved. In fact, it's not as intimidating as it sounds, provided you understand one or two conventions at the start. Once you've got these conventions, the notation in some ways makes things a little bit easier, because now when you meet a new problem and so on, you can commonly guess and correctly guess what the state variables are. But if I take the sloppy picture that I wrote before and write down the energetic scheme more carefully, then I get a representation something like this. I'm going to imagine that there is a variable food. The carbon in the food flows at a rate that is represented by this symbol JEA into reserve, where the total amount of carbon, the total amount of reserve in C moles, if you like, is given by this quantity ME, and it's used for growth that produces structural biomass, whose mass in C moles is this quantity MV, and another fraction of it is used for an endpoint called maturity, which I'll talk about in the second half of this morning's lecture, or for a reproduction. And the relative amounts of carbon assigned to these processes are again given by M with different subscripts. That in turn lets me write down a set of formal definitions, and I've set these out explicitly in this slide mainly for reference, so that if you are looking back through these through these slides, you can see what the definitions are. In the simplest representation, I have 4 variables that characterize the state of an individual organism, what in some ecological literature is called I state, I standing for individual. There is the reserve biomass in C moles, there is the structural biomass in C moles, there is the cumulative reproduction, that's the total amount of carbon that has been allocated to this reproduction buffer by a certain time, and then there is a quantity MH, which is the total amount of energy that has been allocated to increasing a state variable, which I'll talk about later, called maturity. But you can think of it as indicating the state of development, the state of the state of development of the organism. The environment is represented by a variable X, which again I express in C mole units. And then, key to understanding DEB processes is to grasp the concept of flux. And if I go back to the previous slide, the quantity J with a subscript EA here represents the rate at which food is being converted into reserve. You could think of it as the flow of carbon atoms from the food environment into a box labeled reserve. And because of this flow aspect, it's called flux, and we use this symbol JE with subscripts EA to represent the rate at which food is being assimilated into the animal, and that's good unit C moles over time. And then the reserve is used, and I use this symbol J with a subscript EC to represent the rate of mobilization of reserve. And already you're thinking, this is getting crazy. This is a lot of notation. If you look back at this slide, you see that I've actually followed a couple of conventions. A mass in C moles is represented by big M, and the subscript tells you what it is. So this is mass in C moles of stuff. This is mass in C moles of stuff. and so on. So once I've defined a compartment, the symbol big M with that subscript just means the amount of stuff in C moles associated with that carbon, with that compartment. The flux notation is it can be thought of as having two subscripts in just about every case. The first subscript represents the stuff that is being transformed, and the second subscript represents the process. So actually that is a typo. That should be, let me check on that and get back. This one is clear. The rate of mobilization of reserves, that is the flow of reserve due to the process of commitment or mobilization. DEB models also take account of geometry. I've so far presented everything in terms of carbon mass, but just as in my biomass model, I introduced some relationship between carbon mass of an animal and its size, so does DEB. But the key feature of the DEB concept is that the size of the organism is not related to the total biomass, it's related to the amount of structural material. So the assumption is that we can think of a quantity V, which represents the volume of the organism occupied by structure. So in some sense, we're thinking of a space within the organism as being made up of lots of bits of reserve and bits of structure, and some fraction, if you add it all up, is some volume is occupied by structure, and that is what we mean by V. We assume that the length of the animal, what's often called the volumetric length, is proportional to the cube root of that amount of structure. And for making connections with data, we assume that the physical length is proportional to the volumetric length. Because I want to work in carbon-based notation, I introduce a new constant, which in DEB is often represented by the symbol MV with a square bracket round it, and that is the ratio of the actual mass of structure to the volume. And this notation, where a quantity is surrounded by a square bracket, always represents that quantity per unit of structural volume. So wherever you encounter square brackets when you're reading a DEB paper, you think of the quantity inside the brackets per unit of structural volume. And the idea here is that this quantity here, MV with square brackets, for any particular organism, that's intra-specifically, is assumed to have a constant value, and that is, if you like, yet another aspect of strong homeostasis. For many organisms, and the only ones I'm going to talk about in this lecture, it is assumed that shape does not change during ontogeny, during growth. So just as I did with one of the family of models that I talked about last day, I'm going to assume that the rate of assimilation, the JEA, is proportional to surface area. So I make an assumption that the rate at which energy is entering the reserve is some constant here, which represents the maximum assimilation rate per unit of surface area, per L squared, times structural volume to the power 2/3 times a scaled functional response, which is exactly like the phi, the big phi that I had for a scaled functional response in the last lecture. I've tried to summarize this story of notation in these two slides, so you have them for back reference. I realize they are a lot of information to absorb in a lecture, and I'm going to try and minimize the number of equations that I put on the slides while capturing the essential remaining features of the derivation of the model equations. One key assumption in the model is that if I go back to the original model, is that the energy to support all the various processes here comes from the reserves, and we need some kind of rule for partitioning. And this is the so-called Kappa Rule that for many people characterizes the model. Immediately after we break, I'm going to talk you in a little bit of detail through the Kappa Rule and how it works, and then we'll move on and talk about some of the subsequent features of the model, so that by the end of the second part we've reached the stage that we pretty well have the complete model put together. So it's now 9:41 on my computer. I'm going to break for 10 minutes and resume shortly after 9:50 Pacific time. And meanwhile, I'm going to stop the recording. So I'm going to start again, and I want to immediately start by saying that I misspoke when I said there was a typo in a slide. The truth was that my eyesight was playing tricks. The slide was fine. I couldn't read the small image on my computer screen, so there was no problem at that point in the previous session. I'd reached the point in the presentation where I was now talking about the fate of mobilized energy from the reproductive buffer. And the idea in the standard DEB model is that energy from the reproductive buffer, broadly speaking, gets partitioned for two groups of purposes. The first is energy goes to support growth, which is defined as the production of new structural biomass, and for somatic maintenance, which is the term used for the costs of maintaining this structural biomass. The second branch is all the remaining energy, and that is used for two purposes. In embryos and juveniles, it goes to increasing what is called the state of maturity, or, if you like, to support development. And you can imagine that all the way through to reproductive maturity, there is a steady increase in the complexity of an organism, and that is described as a increase in maturity, and it is assumed that has a total energy that has an energy cost. It is assumed that maintaining this state of maturity has a cost, that to just retain a complex structure in some way has costs. But once reproductive maturity is reached, once an organism has reached the adult stage, it's assumed that for all practical purposes development stops. You still have to pay the costs of maintaining this state of maturity, but there is no further increase in development. The energy that for a juvenile is assigned to increasing maturity, for an adult is assigned to reproduction. The key assumption in the model is that a fixed fraction of the mobilized energy goes to each of these groups of purposes. Growth and somatic maintenance gets a fraction which is represented by the Greek letter kappa. The remaining fraction, 1 minus kappa, goes to increasing a maturity or to a reproduction. And although not essential for the theory, and Chapter 2 of DEB 3 elaborates a little bit on this, although not essential for the theory, in virtually every practical application, the quantity kappa is assumed to be constant. But there are exceptions to that. Mathematically, what this amounts to saying, if my pointer is now visible on the screen, is that some fraction of the energy, of the mobilization energy, the quantity JEC in the above picture, goes to maintenance, and that is the flux of reserve that is used for maintenance, this same way of thinking about the symbols that I talked about. And the remainder, this is the flux of energy that goes to growth. If you assume that the cost of maintenance is proportional to the structural biomass, that's very like the assumption I made last week, except there I said the maintenance cost was proportional to the total biomass. Here we're saying it's proportional to the structural biomass. Then we can now say that the flux of energy to growth, the flux of energy reserved to growth, is just kappa times the amount that was mobilized minus the amount that goes to maintenance. And once we've got this, we can imagine that there is some conversion efficiency of a reserve into structure because they are two different types of generalized compound. We introduce a little coefficient in here, which I've represented with a y, and we get an equation for the rate of change of structural biomass. The problem with all of that is that I don't yet have a specified problem, because I started by saying that there was a flux from the environment through the assimilation process into the reserve buffer. I then said that there was a rate of mobilization. I specified very carefully the assumption that related to this JEA, but we don't know what this JEC is. We don't know the rate of mobilization. And this is the single sticking point where many people get lost in understanding the basic DEB model. And I want to talk through, I've got a blank slide here. I want to talk through the logic of that in detail. These slides were because I actually planned to have the break at a slightly different point where I had it. So now, the way we get around the problem that we don't know the mobilization flux is to again recognize that we are not talking about a inert chemical or a biological, an abiotic, that's the word I'm looking for, an abiotic chemical reactor. We're looking at a living organism, and living organism is characterized by homeostatic processes. It regulates the relative amounts of different compounds in it. We are saying here that an animal is made up of primarily of two types of compound, generalized compound, reserve and structure, and it regulates the relative proportions of these. So in terms of the picture, we can imagine that there is a reserve here, and there is some kind of valve represented by this object on my picture that represents the rate at which mobilization is occurring. I introduce a quantity little m with a subscript e, which is the ratio of reserve to structure, and I assume that the organism is regulating reserve density by controlling this mobilization flux. And the question is, what are the rules that are followed by the controller? And the rule followed by the controller is specified in this equation here. And I'm going to do something which might appear surprising to you. I'm going to say remarkably little about where this equation comes from. I'm going to describe briefly its essential property. The property is that the controller has the property that it tries to regulate the ratio of reserve to structure, and it tries to regulate the reserve to structure at a rate that would ensure that this reserve density ME finishes up being proportional to the scaled functional response. You can devote a large amount of energy to understanding the rationale for this particular equation, and it is an interesting and fascinating part of the theory. But for now, I want us to simply argue that there is a regulatory process, that you can make different assumptions about different regulatory processes, but this particular one has a number of desirable properties that are described in Chapter 2 of DEB 3, and I'm going to assume that this equation holds. It introduces two new parameters. One is a quantity called the energy conductance. The other is a measure of the maximum possible reserve density. The section of Chapter 2 of DEB 3 is the single place where people trying to understand the theory frequently get stuck. My advice is, don't worry on a first pass about where this equation comes from or the rationalization. That is for a second pass of the theory when you're thinking more deeply. The important thing is to understand essentially the concept I've put in the picture at the top here, that there is, if you like, a valve and the process is regulated, and this is the equation for the controller. Because if you accept this equation, everything else that follows is just algebra. So if you accept that, I now reach the point that I have two balance equations written at the top of that picture. We have a regulation equation, what I called the equation in the previous slide, the equation that's describing the controller, and we have a definition, which is that the ME was the ratio of the total mass in carbon units of reserve to the total mass in C moles of structure. I can differentiate this last equation to get an equation down at the bottom there, and what I have is four equations in four unknowns. And four equations in four unknowns can be solved. It's tedious and boring, and I don't want to put the algebra on the board, but when you do it, you finish up with a remarkably simple result. If I define a quantity little e to be, again, this ratio of the reserve density to the maximum reserve density, the controller equation that I put down takes this form on the right-hand side of the top line, and the final equation for the growth of structure takes the form that the rate of change of the structural biomass is a pile of things in parentheses here times the rate of change of structural biomass, and the complexity comes from the fact that length is related to structural biomass. So if you express everything in terms of MV, it gets a bit uglier. But if you go further and recognize an assumption that I made just before the break, that there's a relationship between the mass of structure in C moles and length, then we get a very simple equation for the rate of change of length. This simplifies to the form I've put in the right-hand side of the second bottom line in terms of pure length, where there is a constant gamma whose formula I've written down here. And you will feel you've seen this before last week. We're actually back to von Bertalanffy growth. So we build all these relatively complicated assumptions into a model. We do a lot of algebraic juggling, and we're actually back to von Bertalanffy growth. So what's the difference? Let's remind ourselves of the major result from last week. I wrote down the von Bertalanffy growth equation in exactly the same form as on the previous slide. I wrote down a formula for the maximum length, and it was an assimilation efficiency, a quantity theta A, which was an allocation parameter very like the kappa in the Koyman model. There was a parameter related to maximum feeding, and there was a scaled functional response. On the bottom line, there was a maintenance rate, and there was a parameter involving the relationship of mass to length. And the so-called von Bertalanffy growth rate, the quantity gamma, was simply the maintenance rate divided by 3. Let's compare that with Bass's DEB model. The maximum length involves an allocation parameter, a parameter in maximum assimilation. It involves a maintenance rate and a parameter related to length. Very similar to what is up above. But there is a big difference in the von Bertalanffy growth rate. The von Bertalanffy growth rate involves a basal maintenance rate, a somatic maintenance rate divided by 3, just as before, but then it involves a whole collection of parameters related to reserves. So let me just go back one slide, or back 2 slides, and show you. It involved this quantity G, this quantity that I'm now pointing to called G, and the definition of little g involved the quantity mv, involved GEAM. Importantly, it involved the energy conductance, which came from the reserve equation. There was just stuff associated with reserves. And now you suddenly begin to see why I put together in a slide in the first session a picture on von Bertalanffy growth rates and a picture on respiration rate, and said that they were related in some way to reserves. The incorporation of reserves here turns out, and we'll explore that in a little more detail later on in the course, to actually let us say something about interspecific, that's variation between species in rates, both the von Bertalanffy growth rate and respiration rates, which I will be talking about later. I've set this out reasonably carefully in order to emphasize both the similarities and the dissimilarities of the DEB concepts that I've been building up in the last little bit, and what we learned in the previous session by thinking about mass budgets, in order to illustrate the payoff or the benefit for thinking in this somewhat more abstract way about reserves. I now want to explore some of these ideas a little bit further. If I now take the DEB model and I write down the von Bertalanffy growth equation, and I write down the formula for the von Bertalanffy growth rate, and I've stuck with this symbol gamma that I used in the previous lecture for ease of comparison, and I look at the definition of this quantity little g, then I think there is an argument that lets us say that when I look at G, energy conductance is a quantity that came from a basic regulatory or a control equation. We can imagine that that might be broadly constant across species. We could imagine that the quantity mv in square brackets, the carbon mass per unit of structure, when I say broadly equal, I mean within a factor of 4, 5, 6, maybe even up to 10, but not varying by factors of thousands. Likewise, the kappa is constrained in the value it can take, and these conversion efficiencies are maintained. Essentially, we expect all quantities except this one that represents a maximum assimilation rate in there to vary across species, and we can go through a little bit of argument that now says that if we now think about the maximum size of an animal that is getting a maximum food, that is little f equals 1, then this quantity should vary interspecifically with maximum size. And the implication of that is that the von Bertalanffy growth rate should vary inversely with length. The bigger the animal, the smaller the von Bertalanffy growth rate. That is consistent with intuition, but it emerges directly from the theory, and the picture is shown there. If I look at the x-axis, this is the picture you saw before. I have the x-axis representing length in a log scale, the y-axis representing von Bertalanffy growth rate. There is a huge collection of species assembled here. This picture is taken from a paper that Eric Muller, Dina Lika, Bas Kleiman, and I wrote about 10 years ago, but it's actually also pretty much the same picture is in the DEB book. That's growth. I now want to look at the other branch of the picture, which describes the processes of energy allocation to maturity and to reproduction. The portion that is indicated with the box at the bottom right of this figure. When I do that, I need at this point to start thinking more carefully about precisely how we're going to interpret what goes on there. As I've already said informally, I now formalize the fact that juveniles are assumed to use all the material that they get down this branch of the model to increase complexity or maturity. It's an abstract concept that we can debate in detail, but because I want to pull all the bits together from the model in a single session, I'm asking us to take it on trust just now. Maintaining maturity requires some energy, which is called maturity maintenance. Maturity is not something that you measure as part of the mass of the organism. The energy assigned to maturity is assumed to be energy that is dissipated in advancing development. So in terms of the mass balances in the model, it is released as a metabolic product. If we're thinking of carbon as I am through here, it would be CO2 that comes out. The model assumes that adults do not increase their maturity any further under normal conditions, but they do continue to pay maturity maintenance. They pay the cost. They have to maintain their state of maturity. For an adult, mobilized reserves are not required for mobilized reserves that are not required for maturity maintenance are assigned to reproduction. So there's a certain flux, 1 minus kappa times the mobilization rate, that is assigned to a reproduction for adults. From that is debited a cost of just maintaining the state of the animal's maturity, and the rest is allocated to reproduction. This variable maturity is used to determine the point at which the transition from juvenile to adult occurs. So maturity steadily increases for a juvenile when it reaches a critical level called a puberty. By most of the people who work with DEB theory, the animal transitions from a juvenile to an adult, and the adult rules come into play. A side remark on the word puberty: it's very widely used in the DEB literature with a different meaning from the standard English. It has nothing to do with it has no restriction to sexual organisms, and I simply point down at the bottom what I call the Humpty Dumpty principle, which I think applies to a lot of DEB theory. According to Lewis Carroll in Through the Looking Glass, Humpty Dumpty said, when I use a word, it means just what I choose it to mean, neither more nor less. And many DEB concepts have very precise definitions that don't necessarily match the ones that they have in common use English. So just don't forget Humpty Dumpty. Now, I look at this bottom right portion, the portion that was in broken lines in my previous slide, and I ask how I write that down dynamically. I have a flux here, rate of mobilization, a fraction 1 minus kappa of it goes to maturity maintenance or to maturity or reproduction. And the rules that I've written down on the previous slide simply are expressed in math down in the two bottom portions there. The rate of change of the maturity variable for juveniles is given by the expression 1 minus kappa times the mobilization flux minus the maturity maintenance, and there is no change from 0 in the in the size of the reproduction buffer. There is no energy allocated at this point to reproduction, and the roles are reversed with adults, and a single rule for maturity maintenance for both life stages. You can pull all this together in a single. You can pull all this together in a single picture, and you get you get a reasonable representation of what is going on for a feeding organism. And I took this picture from actually a very nice paper by Tanya Souza and I think 3 other co-authors, including Vas Koyman, which is the opening paper in a special issue of the Proceedings of the Philosophical Transactions of the Royal Society that came out about 2.5 years ago. And that model presents that paper presents in a very rigorous and clear way all the assumptions that go into the standard model. I've tried to present them in this lecture in what I would call an inductive way. That's presenting some ideas, some data, and proposing some equations. They've written it down very carefully. It is a superb reference, that paper. And if you're struggling again to try and figure out how all the bits go together, there is a very uncluttered and very readable presentation in that paper. And I, of course, can't present a whole model without letting you know whether my boss, the Daphne, thinks the model's okay. And this is just one example of fitting some data on growth and reproduction with Daphne, and the model does not too badly. I've dropped a series of hints in this lecture that, although I've been talking about growth and talking about vertical growth rates, that we keep needing to think about respiration. And the way I've set up the DEB model using C moles as the currency, it's very easy to think of respiration as CO2 production. We can now ask ourselves, what are the processes in the model that I've put together that will result in the production of CO2? They're listed here. Somatic and maturity maintenance each will lead to CO2 production. There is some overhead associated with the assimilation process, digestion of food, transport of food across the wall of the gut, and so on, all require energy, and there is some overhead. The creation of maturity is a process that leads to evolution of CO2 in this representation, because the maturity state variable has no associated mass. And the idea here is that the energy that would otherwise be going to reproduction is being dissipated as CO2 in the model. And then there are some overheads in converting reserve into structure.
Each of these terms that I wrote down there is a DEB flux, meaning something with a J and two subscripts. And in DEB, the respiration rate is thus a linear combination of fluxes. It's not a fundamental quantity. It's a derived quantity which involves contributions from all these little bits. And that, again, is an idea that we anticipated in the biomass budget model last week. In my model last week, I said that respiration rate was made up of two components. There was the term proportional to the rate of feeding, and there was the background or basal metabolic rate. The same idea is emerging from the full DEB model. It's a much more complicated collection of terms. But in constant food, the DEB model, it's actually well approximated by the same formula. It's essentially a linear combination of a term proportional to structural mass and a term proportional to the assimilation rate. And this is actually consistent with data. Again, I'm referring to the most important animal in the universe, Daphnia, but in the lab of Winfried Lampert in North Germany, a lab that for probably a couple of decades performed very, very detailed studies on Daphnia physiology. One study looked at how respiration rate varied for animals that were being grown in different food concentrations. And it's not a particularly good slide here, but the x-axis represents the concentration of food. The y-axis represents the respiration rate for animals of a given size. And there was a certain minimum, and then as the concentration of food went up, the respiration rate went up and eventually flattened off. Interestingly, the concentration at which, the food concentration at which the respiration rate began to flatten off was very similar to the food concentration at which the functional response flattened off. In other words, broadly speaking, the respiration rate contained a constant background amount and an amount that was related to the feeding rate. And that is exactly what the DEB picture here is trying to capture. And it's not a pedantic matter. In the case we're looking at here, it was something like a doubling of the respiration rate between the least well-fed and the most well-fed animals. And in many cases, the metabolic performance of an animal has a big effect on unobserved maintenance rate. You can exploit this, and this is a theme that we'll be developing in more detail in the next lecture. You can actually exploit the fact that DEB gives you a formula for respiration rate in parameter estimation. The picture here is shows some fits by Bas Kleiman a good long time ago, on where he simultaneously attempted to fit data on ingestion rate, oxygen consumption, growth, and what's called cumulative reproduction, the total amount of energy, or the total amount of eggs produced by a Daphnia over a particular time. And because the same model predicts all these different quantities, it was possible to access a good number of the parameters. And this is another theme that we'll see developing with DEB models, that because a very large number of different phenomena are described by the same, essentially in terms of the same parameters, you can simultaneously try to pull together information from a whole lot of apparently disparate sources and come up with and come up with estimates of model parameters and hence of more predictions. So let me pull all this together by contrasting what we've discussed today with where I got to a week ago. This is the same slide that I ended the opening lecture with. The key components of and what I've listed are the key components of the biomass budget model, and contrasted it and added in red what we've now learned. Carbon balance equations are critical in both. They're just more complicated, or because there's an extra compartment, there are more of them in a DEB. I assumed a carbon weight length relationship, noting that they're often available empirically. In the DEB theory, at least in the standard DEB model, we replace this empirical information by rather careful choice of variables, the reserved structure, and with geometric assumptions. I talked about a maintenance-weight relationship, recognizing that maintenance is never really available directly, and I was kind of sloppy last week in the difference between maintenance and in not bothering to distinguish maintenance from basal metabolism. I noticed that we could get some information from respiration rates at different food levels, but I actually know of very few examples other than the Daphnia picture that I put up where that is available. In the case of the DEB model, we assume that maintenance is related to structure, respiration is more complex, but we have a formula for it. Again, with feeding rate relationships, I was relying on empirical information. DEB makes assumptions that assimilation rate is always related to a surface area, but the relationship between this surface area and structural volume may depend on the shape of the organism, and there's a collection of different shapes. Jargon is V0 morphs, V1 morphs, and so on, that you can read about in the DEB book if you're interested. Both approaches require us to consider a functional response. It's just the same in both, and likewise you can change the rules in both without upsetting anything further downstream in the model. Both require explicit or implicit statements about assimilation efficiency. I finished up very quickly with four possible models for ways of thinking about energy allocation between growth and reproduction in the opening lecture last week. In the case of the model I presented, there was a definite choice in favor of one, given by the Kappa Rule, and that is very, very analogous to what I said about assimilation allocation. So where are we after 2 weeks? Last week I presented what, if you like, was the most intuitive, simplistic, if you like, way of thinking about energy and mass balances and relating them to the physiological performance of a single organism. In this week's lecture, I've pointed a number of drawbacks of the organism. I've tried to take you through, at a reasonable pace, a number of the key steps in understanding how Bas-Kooijman's DEB model is put together in at least its simplest context, where you're talking about an animal. I've pointed out that it immediately gave us a possible approach to thinking about interspecific variation of properties, and I highlighted the role of respiration. And I did that latter because in many ways it's a lead-in to what is going to be the biggest theme of a number of topics that I'll cover in the third lecture, and that is the flows of elemental matter. The fact that although I've written everything down in terms of carbon flow, the model equations that I've written down, combined with the assumption of strong homeostasis, actually lets us say a lot of things about lots of different fluxes.
and I'm going to elaborate on that at the start of the next lecture. So thank you very much for listening, and I hope you're around a week from now. Thank you.